Hi, I'm Liam Ryan. Today I'll present our final report for CS506 on food insecurity and chronic disease. The goal of this project is to examine how food access relates to food insecurity and, in turn, how food insecurity is associated with adverse health outcomes. At a high level, our study follows this straightforward pathway. To explore these relationships, we collected data from the USDA's Food Access Research Atlas, Feeding America's Map the Meal Gap dataset, and the CDC's Places dataset. Rather than merging all the data, we split the pathway into two relationships across 2 years. The first link looks at how low food access relates to food insecurity, using data from 2015 when both the USDA and Feeding America datasets align in time. The second link explores how food insecurity relates to chronic health outcomes, using Feeding America's 2017 data combined with CDC's Places data, which is based on 2017 and 2018 surveys. This setup allows us to examine each step in the causal chain independently, while still getting a broader picture of how structural inequalities in food access may contribute to health disparities. Before splitting the data, we performed exploratory analysis on all three datasets. We identified key metrics for each, for example, food desert population, food insecurity rate, and average chronic disease prevalence, and plotted them as county-level heat maps across the US. What we found was a consistent regional pattern. Counties in the Deep South and Southeast showed low levels of food access, high food insecurity, and high rates of chronic illness. This overlap suggests a potential strong association between these variables, supporting our decision to model these relationships further. After examining overall patterns, we then stratified our analysis by vulnerable groups. Here we map three measures: food insecure children in low income households, children in low access areas, and seniors in low access areas, all at the county level. We see that many counties in the deep south, southeast, and parts of Appalachia have both a high share of low income food insecure children and a high share of children living far from healthy food retailers, suggesting stacked economic and geographic disadvantages. Seniors show a similar pattern in these regions, but also stand out in more rural Midwestern and Western counties, where access barriers appear especially severe for older adults. Another part of our EDA explored demographic disparities in food access using the USDA Atlas data. We computed a disparity index for different groups, including children, seniors, racial groups, households without vehicles, and SNAP participants. The index compares each group's share of the low access population to their overall share in the total population. For example, if a group makes up 10% of the total population, but represents 20% of the population with low access to food, the disparity index would be 2, meaning they are disproportionately affected and 2 times more likely to be in a low access food area. Our methodology for computing this index provides a snapshot at a given distance threshold, so varying definitions of low access can change the results. Overall, the disparity index gives us a quantitative way to identify groups that are most vulnerable to low food access. In our analysis, we observed that Native American populations in rural areas had the highest disparity indices, 25 times higher than the general population. Other groups, like Asian populations in rural areas, were extremely underrepresented. These disparities highlight structural inequalities that go beyond geographic proximity alone. Overall, we were unable to account for all of these demographic factors in our modeling downstream because the Link 2 dataset did not collect the same data. However, we acknowledge that a way to improve this study would be to collect this data across the board and adjust for it. To prepare the data for analysis, we created two clean datasets, one for each causal relationship. Since these sources come at different geographic levels, we needed to standardize them. The USDA Atlas data is available at the census tract level, while the other datasets are reported by county. To align them, we derived FIPS codes from the tract identifiers and aggregated all tract level data up to the county level, using either sums or population-weighted means depending on the variable. This process allowed us to merge everything on a consistent county basis, giving us two ready-to-model datasets, Link 1.csv for food access and insecurity, and Link 2.csv for food insecurity and health outcomes. To quantify these relationships, we applied predictive models for both links. To prepare the data for the models, we trained on 80% of the data and applied PCA to reduce dimensionality. Interestingly, low income, poverty rate, and SNAP status were structural features identified in PCA as consistently correlating with high food insecurity. In both cases, random forests were the strongest performers, with the highest R2 scores, and explained the most variance. They were able to capture nonlinear relationships that linear regression could not. Using our random forest model, we then set out to eliminate features that did not contribute to our model's accuracy. Using recursive feature elimination, we were able to reduce the link 1 dataset from 144 to 46 features. However, when run on link 2 dataset, all but one feature were essential for model fitness. We then ranked remaining features by their importance to the model. In link 1, which predicts level of food insecurity, we found that poverty levels were the biggest driving factor, followed by the share of the Black or African American population living half a mile or more from the closest supermarket. In link 2, we found that childhood food insecurity had the most significant impact on predicting adult chronic illness rates. This correlation could indicate the importance of childhood diet in the development of many chronic diseases. Next, we conducted hyperparameter tuning on our link 1 and link 2 datasets. For link 1, gradient boosting achieved R squared of 0.838, which was a 31% improvement over our 0.64 baseline. This validated that geographic access barriers strongly predict food insecurity. For link 2, gradient boosting reached 0.698, a 9.1% improvement. While more modest, this confirms food insecurity predicts chronic disease prevalence. The key takeaway is that link 1 shows stronger predictive power at 84% variance explained versus link 2 at 70%. Both links validate the complete pathway from low access through food insecurity to health outcomes, providing tools to identify at-risk communities. We tested 3 clustering algorithms to identify natural county groupings: k-means, which was optimal at k equals 2 with a silhouette score of 0.256, dbScan, which produced a negative silhouette score of negative 0.167 and 75 fragmented clusters, which wasn't suitable for the data, and agglomerative, which was the clear winner with a silhouette score of 0.706 using k equals 2 and complete linkage. This high score indicates well-separated and meaningful clusters. The bottom line is that counties naturally fall into 2 distinct groups based on food insecurity and health patterns, enabling targeted intervention strategies for each community type. Next, we used clustering to search for patterns in the data. We tested 3 algorithms, k-means, dbScan, and agglomerative clustering using complete link distance. To evaluate the clusters individually, we looked at silhouette scores, which tells us how cohesive and well separated our clusters are. And to compare them to each other, we used the Adjusted Rand Index, or ARI. All methods place the majority of counties in a single cluster, implying that across all features, the counties are fairly consistent. Further analysis is needed to identify which counties tend to fall into the smaller clusters and why. To explore underlying structure in our datasets, we applied clustering methods to both link 1 and link 2. Across algorithms, we found broadly consistent groupings, but with notable differences. In separation, link 1 shows more overlap between clusters, while link 2 forms clearer, more distinct clusters. Visualizing TSNE embeddings confirm that certain feature combinations, such as poverty rates and food insecurity, strongly influence how counties group together. These results help us see which regions face similar combinations of challenges before moving into modeling. Our 3 key takeaways from this were that clustering can help identify groups impacted by specific challenges concurrently, that what impacts the identity of one cluster may have little to no relevance to another, and that modeling is necessary to capture direct causal relationships. We conclude with the conclusions that chronic diseases and food insecurity are closely linked, influenced by complex factors such as poverty and structural disparities. Through modeling, we can identify high-risk subpopulations with unmet needs and better target interventions. In the future, we'd like to extend analysis to more recent years and evaluate longitudinal trends, examine specific chronic illnesses in greater detail, and incorporate additional datasets to broaden the scope and robustness of the analysis. Thank you for your time.